So, good afternoon. First of all, I have to thank Norberto for bringing me here to a place that I still call home, to speak to a bunch of friends and colleagues and to this nice meeting. My perspective here is my task here is to present you of a new way on bringing together some of the ideas that Norberto just brought with a new perspective of looking into the brain for the last 20 years, that's functional MRI. So before I begin, before I bring this perspective, this new way of looking at these networks, it's important to define more, It's important to define what a complex system is. And because all the strategy that I'm going to follow, it's made off on the complex system framework. So in order to define a complex system, it's important to understand that a complex system is different than a complicated system. So it's important to understand that we are not talking about something that it is indeed complicated. So we have to first understand what is the difference between simple and complicated before we go into the complex system. So something that's simple can be illustrated by a pendulum. A pendulum is a simple system. It's a system that you have the equations that pretty much tells where the pendulum and what the pendulum is going to do. So a pendulum like this one, it's a simple system. A complicated system, on the other hand, so a pendulum is composed based on basically of a limited number of elements. It has very basic laws that are behind its prediction. And on the other hand, a complicated system, like an airplane is composed of a number of pieces. Each piece can be a complicated system itself. Every piece of an airplane needs to work in synchronicity, and it needs someone to operate it. So it needs a central control. On the other hand, a complex system, we can understand a complex system and we can look at a complex system like a bunch of birds migrating. So this is indeed a complex system, a type of complex system. So what it is that defines a complex system? A complex system can be defined basically on these 5 topics. So it has a large number of elements that interacts with each other. It's not guided by a central control, different from an airplane. It presents a self-organizing and collective dynamic. So the dynamic of the system is pretty much defined by how these elements interact with each other. But so they have a self-organizing, they are self-organized. They also present evolution and adaptation different from an airplane. They evolve and they adapt. And the global behavior or the emergent properties that come from the system, they are deducible. They can be deducible from a local analysis. So you can look at the system locally, and sometimes these systems, they express the same behavior if you're looking at them locally or if you're looking at them globally. So on this perspective, on a complex system perspective, it's on this framework, it's where the complex networks appear. So a complex network is basically a way of understanding how these elements on a complex system interact with each other. So it's a way of understanding a global parameter of this complex system. So how do we define a complex network? So we can define a complex network based basically on 2 elements. the first elements are the elements that actually compose the system. So you have a number of elements that you define as being the elements that compose the system, and then for some rules, or for some rule that you define, you are able of defining how these elements interact with each other. So how these elements, for instance, this element here interacts with this one interacts with this one, but this one doesn't interact with this one. And who defines what interaction means is us. So we establish the rule that will guide us on linking different elements. So based on these two definitions, based on the nodes of a complex system and based on the links of a complex system, we can come up with different topographies of different networks. So we can look at, for instance, friendship networks. We can look at how different individuals, how is their relation and taking into account their friendship. We can define a network based, for instance, on actors. We can look at the nodes of the systems then will be defined as different actors, and the links of the systems, they will be defined on how these actors interact. For instance, if they have played a movie together, so that interacts, that makes an interaction of two different elements of the system. So this actor may have acted with this actor in a certain movie, and that established a link between these actors. You can build then a complex system that's based on proteins. You can build a complex system that's based on metabolic reactions, whatever. So basically then, a complex system can be generally defined as a number of elements, a number of nodes, and how these nodes interact with each other. So with this framework in our head, what people have been doing in the last few years is trying to bring this framework into the MRI framework. So it's trying to build these complex networks and understanding the topography of these complex networks based on magnetic resonance imaging. What I'm gonna present is one possibility, one way of defining these complex networks. So basically what most people have been doing, and we have been doing the same thing, is first defining the nodes. So the nodes that we have been using are defined based on some atlas. This is an Oxford atlas that divides our brain into 106 different areas based on anatomy and on these 106 different areas. So this is basically what we have been doing. So we divide our brain into 106 different areas, and each of these areas will constitute a node. So then we have to go for a second step, that is how these nodes are related, how they are linked to each other. One way to do that, and that would make you observe an anatomical network, is based on diffusion tensor imaging. So if basically what you're trying to do with diffusion tensor imaging in this type of work is looking at how those 106 networks are connected with each other. This is not and then you have what is called the structural connectivity network. This is not what we have been doing. We have been looking at a different aspect of MRI. That's what people have been doing that for the latest years. That's using functional MRI, analyzing functional MRI in a different perspective. So when I talk about functional MRI, and most of the speakers that have examples based on fMRI, these examples are a way of understanding how a certain task, how a specific task modulates specific areas of the brain, and then you look at those different areas, and then you characterize these different areas. This is not the strategy that we have been using. We have been using what's called resting state fMRI. So resting state fMRI is something that's a lot simpler than the task-induced fMRI, and basically what you need is having the subject in the scanner for about 5 minutes. The subjects most of the times have their eyes closed and don't have to do anything. So there is a different perspective on looking on how what is the information that these type of exams bring you. It's like when you're doing a task-induced fMRI, you're pretty much studying how your system responds to a specific stimulus. What we have been doing is trying to understand how the system goes on without a specific task. So basically what we have is the ongoing brain activity for 5 minutes. So say that you have an MRI image like this one. What you do for the basic protocol for resting state, the basic image acquisition for resting state is exactly the same image acquisition that you do for task-induced fMRI. So basically what you do is acquire a time series based on volume of your brain. So you sample your brain for a sample frequency of 1 second, 2 seconds, and then you do that along for 3 to 5 to 7 minutes. So basically what you have, you map then each voxel of your brain, you have a specific time series that's related to each voxel of your brain. And with these time series, these are the time series that we use to construct the links, that we use to construct the networks. This is what people have been calling functional networks, in the sense that what you're observing is the natural ongoing neuronal activity when the individual is at rest. On doing that, so basically what you're looking at is how you relate different time series that come from one of those 106 nodes that we already selected based on the atlas. So basically what you do is for each one of those 106 nodes, you have a time series. You have a signal like this one. One strategy on linking two different signals is doing a cross-correlation. And this is the first strategy that most of the people have been using. And that is by doing the cross-correlation between different pairs of nodes, then you're capable of looking at how these two structures, or how these 106 structures interact. And then if they have a higher cross-correlation, that means that those two regions establish a link. If they don't have a higher cross-correlation, they do not establish a link. And by doing that then, what you end up with is with a complex network, like any complex network, that you can try to extract specific information, looking at the brain as a whole, as an entire organ. So you're not looking at the specific nodes of your brain. You're looking at different topographies of your brain under different diseases or under different conditions. For instance, we have used this same framework that I'm presenting to epilepsy, to schizophrenia, and to a psychedelic drug called ayahuasca. We have been using the same thing, and what is interesting is that each one of these three examples, they present highly different topography. Their networks are highly different. So once you have the topography, then you can extract attributes from this network. You can extract information that introduces pretty much the global behavior of this network. And then you can look at different topography. So like Norberto was just telling us, you have a topography that is pretty much like a small world network. You have a random network. You have regular organized network. You have different types of network. One first thing that people have found using this strategy is that our brain, when you build the functional network, and look at the complex networks, like using these strategies, the first thing you see is that our brain, these networks, they are scale-free and they are small world networks. So it's interesting to understand what is a small world network. So basically, when you look at these two different types of networks, so on the left-hand side, you have a regular network. So a regular network, as you can see, is a network where the nodes are regularly connected to their neighborhoods. So you can construct different types of regular networks, and this is one type of regular network. On the other hand, on the right here, you have a random network. So these random networks, they can have connections with neighborhoods, or they can have these connections that are long, they have long connections. These two networks, they have advantages and disadvantages, and one that's very intuitive here is that if you want to send in a regular network, if you want to send information from one end of the network to the other end of the network, that would take a lot of time. The path of this information would be long to reach where you want to go. On the other hand, these are very efficient networks in a sense that they are really well organized and they are really highly clustered. On the other hand, if you go for a random network, you see that in order to establish these long connection, it pays with efficiency of the network. So you have a lot of connections, you have a lot of long-range connections that do, most of the times, they do nothing, and they do not increase significantly the efficiency of information transmission from one end of the network to the other end of the network. So what is a complex network strategy is something that's right in the middle. This network was proposed and further studied by a group by Stephen Strogatz, and the idea behind small world networks is that they're characterized by high clustering, so they are highly connected, but at the same time they have some connections that make shortcuts between one end of the network to the other end of the network. So what is the main example on small world network that we have and that a lot of people talk about is what we call 6 degrees. So basically, this is a pictorial way of presenting the small world network that's the Bacon number. So basically, you can construct a small world network, and this is a small world network, by thinking of an actor or an actress, and think if this actor or actress have acted with Kevin Bacon. And if the actor or actress were in the same movie with Kevin Bacon, then they have a Bacon number of 1. If they act with someone that acts with Kevin Bacon, then that number of that actor is a Bacon number of 2. So if you search for the whole database of the Hollywood actors, you see that the average Bacon number is 2.957. That made a lot of people extrapolate that, saying that everyone in the world separated by not more than 6 nodes. So meaning that I'm connected to the President of the United States for a couple of nodes. I know someone that knows someone that knows him. And that is in average, is not higher than 6. So what's interesting is that this example of an actor network that has a small world network topography is the same type of network topography that we have, for instance, on Facebook. Facebook and friendship, it actually has a small world network. It follows a small world network topography. And in a sense of efficiency, it is an efficient network in a sense of transmitting information. It is an efficient system, and we can see that on Facebook. This definitely is an efficient way of transmitting information. So it spreads very fast, and it reaches very far regions in a very small amount of time. And this type of network, for some reason, is the same type of network that our brain demonstrated. The topography of these networks is the same topography of network that our brain has when we construct a complex network based on those strategies that I've just presented you with. So on epilepsy, as I said, this is one of the strategies that we have been working on. There are only 2 papers that have used the same type of strategies, not actually the same, but what you see is that you indeed have a disruption on the small world network characteristics of the network. So what you see here on the top here, this one here is path length. So the lambda is path length, the sigma is clustering, and the gamma is small world network. So what people have been claiming is this: the example on top is for idiopathic epilepsy, and the one at the bottom is the temporal lobe epilepsy. So for some reason, what these 2 papers point at is the fact that there is a disruption on the small world network topography of patients with epilepsy. What is the type of information that this brings? So, okay, I have a small world network topography that have changed. From one aspect, from more of a philosophical or from a physics aspect, this is already interesting, is in a sense that you have something that's sensitive enough to be changed by a pathology like epilepsy, and change in a way that disrupt, and I'm going to have this word in quotes, disrupt efficiency of the system on transmitting information. And again, I have this word efficiency in quotes. So what is it that we have been, what is our perspective on this type of work? What we have been trying to do is to use the same type of strategy to go for the individual, to try to contribute with a preoperative mapping, a biomarker of preoperative mapping based on these type of strategies. So those 2 papers that I have presented, they are based on average mapping. They are based on the comparison between patients with epilepsy with normal controls. One thing that we need to work on is to try to better understand what these what these changes mean. So, okay, I have a disruption on a small world network due to a decrease in clustering coefficient, for instance. So what is it? How does that translate to a more of a fundamental neuroscience perspective? And in order to gain insights on that, it's certainly fundamental to have a good dialogue between a physicist, neuroscientist, a clinical neurologist, and so on. So this perspective will certainly come from this dialogue. That's indeed beginning on the last few years. And then we need, as a whole, this is something that we have been trying to look for strategies to go for the individual independently on if that has a clinical perspective behind it. I believe that neuroscience will have to change the way we look at most of the data that we have been presenting, because we present our data based on average. And for human beings, particularly, average is certainly not the most fundamental aspects of what define human. Standard deviation is. So in this sense, there are a lot of nice strategies that have been trying to better understand the differences between individuals and not stopping in the average differences between individuals. To finish, I'd like to thank all the collaborators that we have on this work. Part of this work have been conducted in a partnership, in a cooperative project between the Brain Institute in Natal and the School of Medicine here in Ribeirão Preto. So these people come from these two institutions. So thank you so much. Thank you again, Norberto. Thank you for all the organizing committee. I really felt not only at home, but I felt that a lot of people are taking care of me. So thank you so much, and thank you all for your patience.
Thanks, Drol, for a wonderful talk. So as I told, I think we'll be open for questions for Drolio. Maybe we can sit down a little bit there, Drolio? Well, no? Okay. So it's open for questions. I think we have like 10 minutes, something like that, because he won't be with us at the discussion, general. Luisa, there's a microphone. But let me ask a question before the phone. I mean, it's taking profit of the phone with us. I was asking at the end of my presentation, maybe I can ask Drolio on that, how to compose these levels of complexity, how to put together like semiology, which is a phenomenon as an outcome of those circuits, and how to take care of the different locations, the different time courses, because you have time for behavior like an extension in a tonic seizure, whatever, something else. You have the flux, the hyperflux, for example, in a SPECT at some time. You have during some time the functional MRI. So the construction of these networks at some point is not exactly coherent in terms of space and time. However, they are happening in the same individual. So how to place together, because I'm trying to imagine how to put this in, for looking for coherence.
Well, I believe the first strategy that most of the people have been trying is to, at least using the idea of complex networks, is to try to standardize the way that the links are built, in the sense that the links have to translate something that's common between fMRI or semiology or whatever model you're looking at. So, for instance, the type of analysis that we are conducting, it infers a covariance between structures, right? So this is the type of information we have. We have an information that means it's a binding between different structures. So if we want, and if we are to link that to something else, we'll have to go for something that brings the same type of information, because otherwise we're going to keep looking at different topographies of the network. So we can build a network based on the semiology and the links between different individuals on the semiology, whatever you call it, individual, like words or whatever. They have to carry a link in order to understand that as a single phenomena. So I believe that this is one way of doing it.
So basically, I'm proposing a cooperation with Braulio in order to think together. Luisa.
I think the brain is very complex.
I'm sorry?
I think that the brain is very complex.
It is, it is.
However, I think it's more complex than the idea you try to give us.
I'm sorry, the idea to try to?
To give us. you are saying that it's very complex. However, I think it's more complex. How are you going to figure out the plasticity the human brain has?
Right.
The children's brain is different
Right.
to the human brain.
So-
The brain of a person from Japan is completely different to the brain of a person from Mexico or US. Then the complexity is very, very high.
Right, yeah. I'm sure that the type of microscope that we are using to look at the brain is a very bad one, but it's the best we have. So all that we are doing is to use the best thing we have. And by using this very bad microscope, we already, I believe, that we have evolved in better understanding the complexity of our brain. So I'm not saying that this is going to do and resolve all our problems. Definitely not. All I'm saying is that interesting enough is by using this microscope, we are able, for instance, to detect a small world organization of the brain that, and this is something that's interesting, that have been detected in using different microscope, looking at this organ using different microscopes. So all we are doing is looking at it using a specific perspective and gaining insights on these perspectives. There is one thing that there is when fMRI when fMRI started in 1992, so we just made 20 years of fMRI. A lot of people thought that fMRI was a tool that it was so vague, it was so rude, that a lot of people thought, well, this is a technique that won't stand long. It won't stay here for too long. And last year there was a paper on Nature celebrating the 20 years of the fMRI technique. And there is a professor called Carl Friston, who is someone that worked from the beginning of the technique. And he said something that it really touched me, in a sense that he also was one of the people saying that, you know, this won't last long. And the reason why it did is because it's evolving. It's not. So if you think of fMRI a couple of years ago, fMRI was a strategy to do, to revive the phrenology. So there is a nice book called the Neo-Phrenology, and this is not fMRI. Like the blobology that fMRI brought at the beginning, this is not fMRI anymore. So what I'm trying to say is that although we're using this technique, we have been evolving in a sense to extract information that we could not extract a couple of years ago. And using this same type of thing, one thing we have been doing is to look at the temporal evolution of the network and extract information from the temporal evolution of the network and to understand the network as a whole. So I believe that this is also an important perspective that the brain should be looked at.
Good afternoon. Thank you for the presentation. Amazing, really amazing. Well, I have said that through fMRI, we know already 106 nodes, the central nervous system, right? But how far are we, for example, how many connections do we already know among all these nodes, all these brain areas? And how do you think that this can help not just epileptic patients, but already schizophrenias and depression and so on? Thank you.
I believe that the first type of help that we need to do is to understand how it reacts to this global insult. So, like I said, we have something that's sensitive enough to detect changes. So the second step is to understand what it means. So one thing, just to this is not clear enough, but one organization of the network that we have been finding, you can see in this picture that you have networks, you have clusters of networks that are linked by a couple of really small number of links, right? So one thing that we have been seeing is that once you, like schizophrenia, what it does on the network topography is that it increases greatly the path length. So it doesn't really disconnect the whole thing. It doesn't really change the whole network. It does change specific links. It doesn't change the hubs. So this is something that it is as if, one thing that we could speak about is like an airport network. So what was going to happen if the hub, Frankfurt, went down? The whole network goes down, right? So this is one possible disruption. And the other thing is to tell you, well, this is not what's going on. The hub Frankfurt, the hub New York, the hub São Paulo, these hubs, they are all operational. But the link of some of these hubs is broken. So then to go from São Paulo to Frankfurt, I cannot go in a direct flight. I have to fly through New York. And that has an impact over New York. So this is something that, and this is the way we have to evolve thinking. I mean, we have to better understand what are these subnetworks, what's happening with these subnetworks, and I believe that this is the main focus of this type of work.
Hi. Just a single comment on those two questions, just very quick. One of the things in experimental models will be useful for to check the plasticity. So over time, changes in these networks, quite controlled. In patients with unsuccessful surgeries, for example, it's a pre-operation functional MRI and the unsuccessful time, so taking like frames or time windows after, so you can see the difference between the two spots. And the other question is there is already at UCLA a huge amount of information from different patients, not only epilepsy, depression, some others, OCD, etc. You have these networks, you have this anatomical platform. You can sort of play doing surgical experiments in silico. You can sort of, whatever, move from the network, like the temporal lobe, the frontal lobe, the arbitral-frontal cortex, etc., and look at the network with and without some parts you choose to remove, which is exactly what Draulio is talking about. You can sort of play with the networks, with the modeling, predict something, and look at reality if that makes sense or not.
Hi, Draulio. Thanks for the very colorful presentation. At least the images are pretty beautiful. Thank you a lot. My question is a little bit about what you just said, about if you destroy or you block a hub, what happened. So if the epileptic brain, well, let's imagine that as a patient that cannot control his seizures with medication, then these patients go to surgery. You resect a part of the brain. And I was always impressed about the individual that could live with minimal sequela or minimal problems regarding if it take a huge amount of temporal lobe. In your opinion, do you know any study that compares resting states networks before and after resecting as such a huge part of the brain? Is the epileptic brain more complex? The complex networks of the epileptic patient is more complex than a normal patient? Is it more? It's get more complex or get less complex? Do you have any comment on that?
No. Well, yeah, I have comments, but there aren't really any work that have done that, looking at the brain. And there is a technical detail on doing that. That is, you remove a hub that alters. In a sense, the topography of these two networks are different from the start. So there is a technical difficulty on conducting these type of experiments. But there is one thing that you brought that I believe is worth noting is the fact that once you have that brain that's being under an insult for a long time, that brain, and you compare that after resection, the impact on the networks compared to if you were to do a resection on a normal control is certainly not the same, right? So you have something that's been under insult for a long time, meaning that by doing this type of comparison, you are, the type of comparison, I mean before the resection, you are already moving towards where you're looking at. So you're looking at something that, like the talks we had this morning, point to this target, saying that it you know, this is not as functional as it was before. So in this sense,
Professor, thank you for a presentation. I'm thinking about the stability of the topography of this network. If I understand well, the link between two nodes is as strong as the correlation in time of these nodes.
right.
We know from the neurophysiological studies, it's this neural signal is a non-stationary one. So if, for example, instead you get 6 minutes of the signal, you get the first 2 minutes and the last 2 minutes,
Right.
and see this correlation, the topography of the network will be the same, or there is some work about this?
That's a nice question. There are two answers to your question. The first one is that you can look at fMRI data as a low-pass filter. Because what you're looking at the hemodynamic response, so you're looking something on the order of a seconds, what makes the signal much more stable that the electrical physiological data. So this is one thing. The second thing is what you do to circumvent this problem is to analyze it in small beans. So what you have is it's a composite of these small beans networks, and then you average out these small beans. So that made, and so in one sense, what you're doing is the same thing that you do in electrophysiology, is try to reduce the window in order to have it as stationary as possible. So with these two things, then you look at these data, and they are, I'd say, stable, pretty much stable.
Yeah, you just continue this question. In terms of neurodynamics, I mean, you're building a map that started out by the CITO architecture, based like a Brodmann map or associated or not to function. And then you use the DTI imaging to study a little bit of connectivity. But then again, you can have the same areas being recruited in covariance and having completely different functions.
Right.
So the whole idea is that these, the connectivity and the nodes themselves might actually change drastically depending on the situation, depending on the context, depending on a lot of things.
Right.
So in that sense, you would have several maps for several circumstances.
Right.
And you got to remember that in terms of epilepsy, it's normal until it isn't. Mm-hmm. So the whole idea is that you can have this idea of okay, I blew up a specific bridge, so I have to go to New York to go to Frankfurt. But then again, other dynamical pathways will appear in that process. So you cannot maintain the structure of the idea. So how would you deal with that?
That's a great, Márcio. One thing that we have to understand here is that we're not looking at specific nodes. So although you are correct saying that you can go either from São Paulo to Frankfurt going through New York or from Milan, what happens is that you increase the path length. So in a sense, what you're looking at, it's a global feature, a global characteristic. So you're not looking locally. It doesn't matter if this and this, so it doesn't matter 1 and 2. It matters the global properties of your network. So what we are seeing is that the global properties of the networks, they are stable. In a sense, for instance, if I map most of us in the room, and I do a resting state of most of us in the room, our networks will be small world networks. So in this sense, we are looking at the global properties of the network and not at local. So we are different. Although we are different, the global structure, the complex network that underline my brain and your brain follows the same topography. This is what we have been looking at. So it's not really a local property. It's a global property.
Can I ask one question?
Oh.
Hi,
Gabriel. Hi.
So can I add to Marcio's comment here?
Sure.
What I believe he was saying is that you can take a plane and go to São Paulo and Frankfurt, or you can take a plane and go to Addis Ababa, to, I don't know, to Johannesburg,
Right.
and it will be the same because it's just a connection. 2 points, 2 airports, ticket plane, and you go. He was affirming that in the brain, not all connections are created equal. So that's the big difference. So if we compare here with the human network in this room here, would be almost the same thing. Who has Facebook friend with who here?
Right,
we are
right.
comparing here and saying how many nodes you need
Right.
to jump from one person to another.
Right.
So Facebook links will be the same. You're a friend and that's it. But not all connections are created equal. You can
Right.
be a sister or a mother or a daughter or a cousin or a, I
Right.
don't know.
Right.
So even in human relations, we have this hierarchy of the connections. And in
Right.
the brain, for sure, it's like that.
Right.
So if you have a thalamocortical connection, it's different to have a hippocampal subiculum para hippocampal gyrus. So you can, that's the problem, that if you map the connections, okay, it's great, it has a big value. And with that result, you have shown that you can map and locate the seizure focus maybe. But not all connections are created equal. So
Right.
you have to take this into account of the models. Then I think will be great.
Right. Again, Gabriel, this really, for this framework, this is what it's beautiful about this. This doesn't matter. For this network, it doesn't matter. For the topography, it doesn't matter if you have, what matters is the whole networks of friendship of the world follows a specific topography. So again, I'm not looking at individuals. So it's a different perspective, really. It's not on the perspective of how you don't weight individuals in these networks. You can, but you're not saying that this particularity of this network is what matters, is what we are looking at. So we are looking at, so what is beautiful behind this is the fact that they share common properties independent on the individual, and this is what defines a complex system. It doesn't matter if that bird, it's the mother of this bird, it doesn't matter. What matters is migrating. This is what matters. This is the complex system. So in this sense, this is what I found most intriguing behind this kind of approach, is that you are not looking at the specificity of individuals, of connections. You're looking at global properties of the network. Again, the birds migrating. This is what matters. Individuals don't matter.
Any additional questions?
Individuals don't matter is strong. That's not what I meant.
But Vitor, to close before the before the coffee break. I think that that's the spirit of that use of computational tools. You can do it. You can think about it. If it explains the mechanisms or not is a question of either going to this clinical setting, if that's the case, going to the bench, or doing another algorithm that proves or not, whatever. So that's creative thinking. So the use of computers in the way we use today, the use of machines like fMRI machines, as we do today, wasn't possible, I can say, 5 years ago. So having that as a tool for thinking, for creative thinking, is wonderful. But I think any following second will be a different network because it's dynamic. But at the same time, we have predictions. We are doing something even worse. We are removing the temporal lobe without those tools. I mean, so we remove the temporal lobe in AH patient in the 50s. We learn a lot about memory from him. So what about the questions on that case in the 50s? We learn about the homunculus brompenthil in the 50s, doing electrical simulation of the whole brain, asking for eloquent areas. So I think we are moving on. We are having more tools, and probably we'll have better options or better use of those tools if our brain is being used appropriately. So I think we are here exactly to share that different point of views and sharing our competence and our experience. Okay, so let's go to the coffee break. Thanks.